most of our knowledge about the human brain has come from the study of a mouse brain. Okay, so this is the line, okay, or the contention which is being given by the author, that most of our knowledge about the human brain has come from the study of a mouse brain. Okay, a neuroscientist dissects mouse brains. Okay, so what is dissects is that it cut it open. Okay, cut opens the mouse brains, hoping to find out more about their major regions and important structures associated with mental and neurological disorders such as autism or epilepsy, and how to fix them. Okay, so what this author is saying is that most of our knowledge about the human brain has actually come from the study of a mouse brain. Okay, and neuroscientist cuts mouse brains, okay, hoping that it is going to find out major regions and important structures associated with the mental and neurological disorders. Okay, and how to actually fix them. The knowledge so gained is then applied to treat human patients, because the human brain is quite similar to the mouse brain. Okay, so that is the reason that most of our knowledge about the human brain has actually come from the study of a mouse brain. Okay, however, these two species have now, however, this is an important idea where the author is getting involved. Okay, so however, these two species have evolved in different ways, adapting the brain of each of their respective lifestyles and evolutionary niches. Okay, so no doubt that the most of our knowledge comes from the study of a brain, but there are some kind of a differences because they have evolved differently. Okay, they adapt to the lifestyle and the evolutionary niches separately. Okay, this explains why cures in mice don't always translate into cure for humans. Okay, indeed, our reliance on the mouse has left us with many unanswered questions, such as when the first human neuron fires. Okay, so there is some limitation. Okay, so what is this particular passage is saying that the one knowledge about the brain comes from the mouse brain, but lately we realize that the mouse and the human has developed or evolved in totally different ways. Okay, so these studies cannot directly be, so whatever the studies or the findings that we have found related to the mouse brain cannot be directly replicated to the humans. Okay, so in case if I have to summarize this particular paragraph, okay, then I would say that the research basically which is being done on a mouse, okay, so the research which is being done on the mouse, okay, it cannot be replicated. Okay, so a mouse, it cannot be replicated to humans. Okay, because they are slightly different. Okay, so let's move to the second paragraph. Okay, so while we normally use non-invasive techniques such as ultrasound to visualize the developing fetal brain, okay, we can't experimentally investigate it at a resolution that allows us to understand it. Okay, so what is this? Okay, so what is the contention? So right now you can figure out that the idea is related to somewhat related to the understanding of the mental and the neurological disorders, okay, such as autism or epilepsy. Okay, so while we use non-invasive techniques, so what is a non-invasive technique is that basically you don't cut it open. Okay, so while we normally use non-invasive techniques such as ultrasound, so in case if you have ever undergone through the ultrasound, you know that you won't get hurt. Okay, and they don't prick you. Okay, something, so that's the reason that it is a non-invasive technique. Okay, to visualize the developing fetal brain. Okay, now what is a fetal? Okay, so a baby which is in the womb of a mother, okay, that is actually a fetus. Okay, so while we normally use non-invasive techniques such as ultrasound to visualize the developing fetal brain, we can't experimentally investigate it. Okay, so we can't do any kind of an experiment in it, because we can just see that whether the brains has the particular nerves and all that. Okay, whether they have the three parts or not. Okay, so that is what we can see through the ultrasound, but we cannot experimentally investigate it. Okay, at a resolution that allows us to understand it. Okay, it is not possible to invasively study a it is not possible to invasively study a human embryo inside the womb. Okay, so it is not possible to invasively study a human embryo inside the womb. Okay, see what is the author is saying. See, this Down syndrome, this autism, this epilepsy. Okay, this is a genetical disorder, okay, which is there when the baby is in the womb of a mother. Okay, but since we don't have any invasive technique, okay, and we use non-invasive techniques, so it does not hurt the baby. Okay, so it is not possible to actually investigate it. Okay, so that is what the author is saying, that it is not possible to invasively study a human embryo inside the womb. Okay, so not a very like a very informative passage, but just a simple idea, okay, that it is not possible to study brain of human embryo invasively. Okay, so maybe this would be the idea, that it is not possible to study a brain of human embryo invasively. Okay, so that's what I would say is the key idea here. Okay, neuroscientific research would gain much if researchers could study a human brain that is not inside a human being. Okay, so neuroscientific research would gain much if researchers could study a human brain that is not inside a human being. Okay, so in case if I can somehow, in case if we can devise some kind of a technique through which I can study the human brain, okay, which is inside the womb, okay, without actually taking the brain outside, okay, in case if we can pick out some strategy, okay, then the neuroscientific research would gain much. Okay, fortunately in 2008, okay, so that means what does this conveys, that fortunately that something like this what we are expecting actually happens in 2008, when the Japanese researcher Yoshiki Sasai discovered that it was possible to cultivate artificial human neural tissues outside the womb. Okay, so it's possible, okay, that you can actually cultivate the artificial human neural tissues outside the womb. Okay, so called pluripotent stem cells. So what are they technically known as? A pluripotent stem cells. They have the ability to generate cell types of all, for all tissues in the human body. Okay, so they can actually generate the cell types, okay, for all tissues in the human body. Okay, so one structure that you should know is that the cells actually form the tissue and the tissue actually forms the organs. Okay, so that is the reason. So we're talking about the brain as an organ. Okay, so what does this what this contention is all about? Okay, that to understand the development of a fetal brain, okay, scientists has to actually come up with a way. Okay, and then this guy, this Yoshiki Sasai, actually discovered that it is possible to cultivate a artificial human neural tissue which is outside the womb. Okay, and they are technically known as the pluripotent stem cells because they have the ability to generate cells type for all tissues in the human body. Okay, you can get pluripotent stem cells from the human embryo or by reprogramming cells from a more developed organism. Okay, so what you can do is that you can actually extract this pluripotent stem cells from the human embryo, and then in a particular condition you can see, okay, that how this particular stem cells are actually growing, and then you can actually conclude that how the brain is actually growing inside the baby who is inside the womb. Okay, so you can get pluripotent stem cells from the human embryo or by reprogramming cells from a more developed organism. Okay, Sasai has shown that one can guide these cells independent of their origin to become a neural tissues by exposing them to specific external factors. Okay, so you can guide them to become a neural tissue by exposing them to a specific external factors. When suspended in liquid, these neural committed cells spontaneously self-aggregate and form small organized three-dimensional clusters that resembles the anatomic structure of the fetal human brain. Okay, we could call these structures brain organoids or mini brains. Okay, so yeah, so what is saying that initially they start off by saying that it is not possible to study brain of human embryo invasively. Okay, but then there is a scientist, okay, he tells that to understand development of a fetal brain, a scientist named as Yoshiki Sasai, okay, he has devised a way to cultivate these cells outside the womb. Okay, now these cells are called pluripotent cells, okay, which makes complex fetal brain structures called mini brain or a brain organoids. Okay, so if you can guide these cells to become neural tissues by exposing them to the specific external factors. Okay, so that is what overall an important passage. Okay, the most important para I would rather say. Okay, so the brain organoids can now grow up to 0.5 centimeter, a pea-sized body of white tissue that you can see with the naked eye. They are kept floating in a juicy reddish stew that contains all the nutrients the cells need to survive, and indeed they can survive for weeks. Okay, so it is there in the cells need to survive. So cell survival, they can survive for a week, months, and years. Okay, some have even survived 3 full years, and it seems they could be kept alive even longer than that if someone replaces the juice continuously. After keeping brain organoids alive for several months, scientists observed that spontaneous emergence of brain oscillatory waves in them similar to those detected by electroencephalograms. Okay, so now what is this saying? That after keeping brain organoids alive for several months, scientists observed the spontaneous emergence of a brain oscillatory waves in them. Okay, so in case if you keep them alive, okay, so you can see the kind of the waves that is being generated by the brain. The same kinds of the brain waves are being generated by these organoids. Okay, these oscillatory waves are ubiquitous in all living human beings. Okay, so ubiquitous is kind of present everywhere. Okay, present in every human beings, but were never before recorded emerging from any living human brain system. In other words, this simplistic model of the human brain could in some ways recreate the organized frame formation of networks that arise in the womb. Okay, so this organized, okay, it can kind of recreate the organized formation of the networks that actually arise in the womb. Okay, so what this particular paragraph is saying is that there is a possibility or potential in the whole model or the study that you can create similar kind of networks of brains that happen in the womb. Okay, so it is a kind of compensating for the invasive study. Okay, so overall, in case if I have to summarize this particular paragraph, so that's a these rather let me just get into this mode. Okay, so these brain organize, they actually kind of emerge these oscillatory waves, okay, which is similar to human brains, okay, and can help study the formation of the network that actually arise in the brain. Okay, so that is the case. So it's again kind of, let's say, giving a support to the study of a Yoshi society. It should not be forgotten that the brain organoids aren't actually miniaturized human brains in a dish. Okay, so brain organoids are not actual human brains. Okay, they are just the mimic of it. Okay, so nonetheless, this reductionist human brain model was crucial in helping to figure out that the Zika virus was the culprit behind the outbreak of congenital brain defects in the northeast of Brazil in 2015. Okay, so the organoids are not actually brains, but a brain model that helps scientists discover that the Zika virus was the culprit behind the congenital brain defects in the Brazil. Okay, so overall, an easy paragraph, easy passage to read. Okay, and the questions are also easy. Okay, no doubt that stem cells, this organoids and all that, there was some kind of a scientific jargons which were used, but yeah, they were still understandable from the basis of the passage because the author actually tries us to explain them. Okay, now according to the passage, which of the following is not true about the study of mouse brains? Okay, so one thing is actually clear is that the mouse brain was discussed in the first paragraph, so the answer is to be something based on the first paragraph. So according to the passage, which of the following is not true about the study of mouse brains? Okay, so it has allowed us to know the basic features of human brains. So yeah, this is true. Okay, it's written here. Okay, that most of our knowledge about the human brain has actually come from the study of mouse brain. Okay, so we can say that it has allowed us to study the basic features of human brains. Okay, now somebody might raise the question that where it has mentioned that the basic features. Okay, but it is very unlikely that we have learned only the advanced concepts through the study of mouse brain. Okay, in case if we have just learned the advanced concept, then what is the point of actually getting into this development of the brain organoids and all that? Okay, so we can anyhow infer. Okay, so the statement can be considered, or it can be implied by the passage that it has allowed us to know the basic features of the human brain from this line, that most of our knowledge about the study of human brain come from the study of brain. So the mouse brain has actually allowed us to know the basic features of human brains. Okay, it has not expanded our knowledge of the human brain as much as we would like. Okay, yeah, that is the reason. Okay, that is the reason. Okay, because it has not actually that is why a lot of things you are testing on a mouse brain is not getting replicated to human brains. Okay, see, there is a line, no, so this explains. Okay, so however, these two species have evolved in different ways. This explains why cures in mice does not always translate into cures for humans. Okay, indeed, our reliance on the mouse has left us with many unanswered questions, such as when the first human neuron fires. Okay, so since it does not always translate, so cures in mice does not always translate into cures for humans. Okay, or our reliance on mouse has left us with many unanswered questions. Okay, so we can say for sure that it has not expanded our knowledge of the human brains as much as we would like, because we are still seeking for the questions for the answer of these unanswered question. Okay, that is why there are a lot of things that you are testing on a mouse brain is not getting replicated to the human brain, and there are many questions that are actually unanswered. Okay, so we can say that it hasn't expanded our knowledge of the human brain as much as we would like. Okay, it should not be replaced with the study of the brain organized. Okay, so where did the author says that? And this is actually the correct answer. Okay, so according to the passage, which of the following is not true about the study of the mouse brain? It is saying that the mouse brain should not be replaced with the study of brain organized. Okay, nowhere in the passage the author actually says that the brain organized should actually replace the study of the mouse brain, or it should not. Okay, so this is something that we cannot say. So this is something which is not true about the study of the mouse brain. Okay, and secondly, in case if you want to go for a very far-fetched inference, okay, so we can say that because the mouse brain study does not give us a comprehensive study of the human brain, because there is many questions which is unanswered. Okay, and we cannot actually use the invasive technology. Okay, we need to study the brain non-invasively. Okay, important to study the brain, we cannot use the invasive techniques to study the brain, and it is important to study the brain. Okay, so there is a possibility that mouse studies kind of gives us the that you should that this study should be replaced, or this study should be replaced by the study of the brain organized. Okay, but anyhow, let's not get into that technicality. That was just a one kind of a view that I want you to explore. Okay, so it should not be replaced with the study of brain organized. Nowhere the author says that. Okay, so author says that the brain organized is kind of an advanced thing. Okay, but it should replace or not mentioned. Okay, secondly, yeah, there are some kinds of limitations to the mouse brains. Okay, but again, so brain organized is kind of a better version, but it should be replaced, again cannot be commented. Okay, it can help neurologists in developing cure for human brain ailments. Okay, yeah, so this is mentioned here. Okay, however, these two species have evolved in different ways, adapting the brain for their respective lifestyle. This explains why cure in mice does not always translate into cures for humans. Okay, so why it does not always translate? So by this line, what we can infer is that this means that the cures in mice at least sometimes translate into cures for human. Okay, can we infer that? Okay, so it can help neurologists in developing cures for human brain ailments. Okay, because that is a reason. Okay, that we are studying, we are saying that the mouse brain is actually similar to the human brain. Okay, and the knowledge gained is then applied to treat human patients. Okay, so we can say that it can help neurologists in developing cures. Okay, and again, this explains why cures in mice does not always translate into cures for human. So that means that there are some times where the cures in the mice is actually can be translated into cures for humans. Okay, so the fourth is true about the study of a mouse brain. Hence, the 3 is our correct answer. I hope you got the logic. Okay. Now, according to the passage, brain organized helps scientists in all of the following ways except. Okay. Now, whenever there is an except kind of a question, you can actually change the format of a question. Okay. So you can say that according to the passage, brain organized do not help scientists in all of the following ways. Okay, so we have to look for the option where the brain organized do not help scientists. Okay, so brain organized in investigating human brain development and a brain structure, this is true. Okay, it actually, the brain organized actually helps us in investigating human brain development and the brain structure. Okay, so can we see that? Yeah, in other words, okay, this simplistic model of the human brain could in some way recreate the organized formation of the networks that arise in the womb. Okay, so we can say that it actually, the brain organized helps in investigating human brain development and the brain structure. Okay, because the simplistic model of the human brain could some way recreate the organized formation of the networks that arise in the brain. Okay, so we can investigate the human brain development and the brain structure. So not our answer. In obtaining a brain that was never inside a human being. Okay, yeah, this is also true. Okay, that this organoids actually helps us in obtaining a brain that was never inside a human being. Okay, see, neuroscientist research would gain much if researchers could study a human brain that is not inside a human being. And fortunately, in 2008, the Japanese researcher Yoshiki Sasai discovered that it was possible to cultivate. Okay, so this is again true. Okay, now in studying the impact of lifestyles and evolutionary niches on the brain. Okay, Now this is actually a correct answer. We cannot say this thing that the brain organized actually helps in studying the impact of lifestyles and evolutionary niches on the brain. We cannot say that. Okay. In fact, this is the kind of the option which is true as per the passage, but not correct answer. Okay. So this is a line which is mentioned here. Okay. Um, yeah, so adapting the brain, okay, for their respective lifestyle and evolutionary niches. Okay, so this is mentioned to show the difference between a human brain and a mouse brain. Okay, so lifestyle and evolutionary niches, okay, cannot be studied, okay, even in brain organized. Okay, or the author is nothing commented over the this thing. Okay, or maybe we can say that it should not be forgotten that the brain organized aren't actually miniaturized human brains in a dish. Okay, so maybe we cannot say with a certainty that organoids actually helps in studying the impacts of lifestyle and evolutionary niches on the brain. Okay, so that's one of the way. Okay, so this is our answer. Okay, because this was said in terms of the comparison between the mouse brain and the human brain. In studying the development of the brain related diseases, yes, the brain organized actually helps us in studying the development of the brain releases diseases. We can find out here. Okay, so the example that nonetheless this reduction of human border was crucial in helping us figure out that Zika virus was behind the breakdown of congenital brain defects. Okay, so it helps in studying the development of the brain related disease, but we cannot say anything about the option number third. Hence, third is my correct answer. Okay, now according to the passage, Yoshiki Sasai discovered that. Okay, so it is, what does the Yoshiki Sasai discovered that? Okay, so Yoshiki Sasai was kind of mentioned in the third paragraph. Okay, so let's see the third paragraph, that fortunately in 2008, the Japanese researcher Yoshiki Sasai discovered that it was possible to cultivate artificial human neural tissues outside the womb and all that. Okay, okay, let's see. Pluripotent stem cells have the ability to generate cells type for all tissues. Okay, now this is a one good question. Okay, why this is a one good question? Yeah, no doubt that it's a very factual question. It's a direct question. Okay, but it's still tricky. Okay, why? Can you see? Let's read this particular para 3. So fortunately, in 2008, the Japanese researcher Yoshiki Sasai discovered that it was possible. So let me just give you a hint here. Okay, so fortunately in 2008, the Japanese researcher Yoshiki Sasai discovered that it was possible to cultivate artificial human neural tissues outside the womb. Okay, now there is a line which is telling you so-called pluripotent stem cells have the ability to generate cells type for all tissues in the human body. You can get pluripotent stem cells from the human body or by reprogramming cells from a more development organism. Okay, now Sasai has shown that one can guide these cells independent of their origin to become a neural tissue by exposing them to specific external factors. Okay, so when suspended in liquid and all that. Okay, so see, can you see the two underlying part? Okay, so what is the problem and what is the trick here is that this is that pluripotent stem cells has the ability to generate cell types for all tissues. Okay, so it's mentioned that the pluripotent stem cells have the ability to generate cell types for all tissues in the human body. Okay, but the question is that is it is it the thing which is being discovered by the Yoshiki Sasai? Okay, now this is to actually, this is present in a very general format. Okay, this is not being mentioned by the Yoshiki Sasai. So what was being discovered by the Yoshiki Sasai is that Japanese researcher that it was possible to cultivate artificial human neural tissue outside the womb. Okay, now so-called pluripotent cells, it is explained for the reader's benefit. Okay, the discovery is not attributed to the Sasai. Okay, what he told is that it was possible to cultivate artificial human neural tissue. Okay, but they have the ability to generate cells type. This is general information. Okay, not something which is being discovered by Yoshiki Sasai. Okay, now reprogramming cells from a developed organism can make them pluripotent. Now this is again line. Okay, or by reprogramming cells from a more developed organism. Now this is again presented in general manner. Okay, it is again explained for the reader's benefit. It is not attributed to the Sasai. Okay, and one thing you can case, okay, in case if you feel that the pluripotent stem cells have the ability to generate So why not this could be the answer? Okay, so these two cannot be the, since one cannot be the answer, so there's no logic that the second can be the answer, because both of them are presented in a similar manner. Okay, now human neural tissues can be cultivated outside the human body. This is absolutely correct. Okay, because the Japanese researcher Yoshiki Sasai discovered that it was possible to cultivate artificial human neural tissues outside the womb. Okay, so outside the womb and outside the human body. Okay, so this is true. Okay, and directly mentioned, and there is no controversy here. Okay, that mature cells of any origin can be converted to an embryonic like state. Okay, so this is kind of a rephrasing of an option number 2. Okay, which says that you can get pluripotent stem cells from the human embryo, or by reprogramming cells from a more developed organism. Okay, so more developed is the mature cells of any organism. Okay, can be converted into embryonic like state. Okay, so that is again kind of these the lines which is being presented here. Okay, so it's again a kind of a rephrasing of option 2. Okay, so it's a rephrasing, so 2 and 4 cannot be the answer because both of them cannot be my answer. Okay, the one is again present in the similar manner as 2 and 4, whereas the third is the option which is directly present. Okay, so don't get tripped, it was an easy question, but again it was slightly tricky. Okay, last question for this particular passage. Now, this was the most difficult question. So, all of the following, if true, would cast doubts on the hope that human brain organoids would recreate the organized formation of the networks that arise in the womb, except. So, again, except question. So, all of the following, if true, would not cast doubt on the hopes that human brain organized would recreate. So, there's a line, no? In other simple words, this simplistic model of the human brain could in some ways recreate the organized formation of networks that arise in the way. Okay, so all of the following will call cast out, but there is one option which does not cast out. Okay, so let's understand this particular line. Okay, so what does the passage explain? The passage explains that human organoids are actually kind of a mini brains or the tiny brains that can grow outside the womb. Okay, by using stem cells. Okay, which are capable of let's say the stem cells, this is mentioned no here. Okay, so okay, stem cells, yeah, so stem cells which have the capability or the ability to produce any types of tissues. Okay, including the neural thing. Okay, including the neural tissues. This makes it possible, okay, to observe the formation and development of a brain. Okay, without the invasive process. Okay, hence the hope that organoids would recreate the organized formation of networks that actually arise in the womb. Okay, so would cast doubt. Okay, so let's see the first option. Human brains organized don't accurately capture all aspects of development in a brain structure. Okay, so in case if the human brain are organized, they don't accurately, okay, captured all aspects of development in the brain structure. So if all aspects are not accurately captured, then how we can say that we can actually recreate the organized formation of networks that arise in the brain? Okay, how we can say that it can actually make it possible for us to observe the for make it possible for the scientists, actually not us, okay, to observe the formation and development of a brain. Okay, when it cannot accurately capture all aspects, so it actually casts a doubt. Okay, and we have to look for option which does not cast a doubt, hence it's not my answer. Organize like the environmental and lifestyle cues that are essential for brain development. Okay, so organoids lack the environmental and lifestyle cues. Okay, so this is a critical reasoning question. Okay, so the hypothetical question, so don't get confused between this question and this question. Okay, so organoids lack the environmental and lifestyle cues that are essential for brain development. Okay, so this is saying that these two cues are essential for the brain development. Okay, so in case if essential components are lacking, okay, then the development is not necessarily the same. Okay, if organoids cannot modify itself according to the lifestyle and the environment which is essential for the brain development, okay, how we can say that they can actually recreate the organized formation of the networks that arise in the womb? Okay, because they cannot actually they are susceptible to it is saying that the brain development is actually susceptible to the environment and the lifestyle cue. Okay, whereas the organized is not, so it cannot recreate the same kind of a thing. So it's again casting a doubt. Okay, now organoids alter the patterns, so alter the pattern and lack the cellular diversity found in a normally developing brains. Okay, so now this is again altering the pattern. They change the pattern. Okay, so how the brain works in a normal human beings, they actually change the pattern. So if they alter the pattern, it is a more serious problem. Hence, recreation is not possible. Okay, and it is saying that organs alter the pattern and lack the cellular diversity found in a normal developing brains. Okay, if it is different from a normal, if the organs are different from a normally developing brain, then what is the purpose of these organs? Then what is the purpose of this entire study? Okay, if they're totally different from a normal developing brain. Okay, so this is again casting a doubt, hence not by answer. Okay, so by the option elimination, you can see that the fourth is the correct answer. But let's again figure out. Okay, organoids developed outside the womb are open to manipulation of brain cellular process. Okay, so the organoids which are developed outside the womb, okay, they are open to manipulation. Okay, so whatever the manipulation, whatever the brain cellular process is happening, okay, inside the human brain. Okay, so they are actually open to manipulation, that we can actually manipulate them. Okay, according to the brain cellular process. Okay, so in case if we can manipulate them, okay, they are open to manipulation, then it does not cast a doubt and a hope that it is possible to recreate the formation. Okay, so we can manipulate, so we can actually recreate. Okay, so worm-like structure is susceptible to the manipulation of a brain cellular process. Okay, and in case if we can actually do all these kinds of manipulations in the organization, in this organoids, then it is actually, it can create the organized formation of the network. Henceforth is my answer. Okay, so by this we are done with the passage, and I